Hello everyone, and welcome to today's presentation. I'm excited to share with you our insights on the production, extraction, and purification of enzymes. The session is titled Production, Extraction, and Purification of Enzymes, and it's part of the BTC3101 Enzyme Technology course. Over the next few slides, we'll explore how enzymes are made, extracted, and purified for various applications. So let's get started. At the end of this topic, you should be able to identify the suitable process to produce, extract, and purify the enzyme. We'll also cover the techniques involved in each of these steps. This is important because selecting the right process ensures we get a high yield of active enzyme with the desired purity. Throughout the presentation, we'll explore how these methods connect to overall enzyme production goals, helping us understand how to optimize each stage for better efficiency and effectiveness. Now, let's take a look at the typical workflow involved in the production, extraction, and purification of enzymes. First, after obtaining the biological source, whether it's microorganisms, animals, or plants, we start with the initial step, cell disruption. This can be achieved through various methods like grinding or other disintegration techniques to release the enzymes contained inside the cells. Once the cells are broken open, we move on to separation processes such as centrifugation and filtration. Centrifugation helps us separate the heavier cellular debris from the liquid containing our target enzymes, while filtration further clarifies the extract by removing smaller particles. Next, we often perform extraction, which involves isolating the enzymes from other cellular components. This step may include additional purification techniques, like precipitation or affinity chromatography, to increase enzyme purity. Finally, the concentrated enzyme solution is subjected to drying to remove excess moisture, ensuring stability and ease of storage. Sometimes, further purification steps like additional centrifugation or filtration are necessary to achieve the desired enzyme purity for industrial applications. Throughout this process, understanding the distinction between extracellular enzymes, which are secreted outside the cells, and intracellular enzymes, which require cell disruption to extract, is crucial. This entire workflow highlights the importance of efficient separation and purification techniques to obtain high-quality enzymes for various biotechnological uses. So let's talk about the major factors that influence how we choose the techniques and methods for isolating enzymes. One of the most critical factors is the source of the enzyme itself. It could come from animals, plants, or microorganisms, and each source has its own considerations. If we're dealing with plant or animal sources, it's important to identify which tissue or organ contains the highest enzyme activity. For example, certain tissues might be rich in specific enzymes, making them ideal starting points for extraction. Another key point is how we handle these sources before extraction. Ideally, the material should be fresh and kept at low temperatures, around 4 degrees Celsius, if there's a delay before extraction. This helps preserve enzyme activity. Some materials can even be frozen if needed, which allows for longer storage without significant loss of activity. Now you might wonder, what happens if we keep enzyme sources at room temperature? Well, enzymes are quite sensitive. Exposure to higher temperatures can cause denaturation, leading to a loss of activity and making extraction less effective. So, maintaining proper storage conditions is crucial for successful enzyme isolation, and choosing the right source based on enzyme activity and tissue type is a fundamental step in the overall process. Let's talk about the location of the enzyme and where it's typically found. Enzymes can be intracellular, meaning they are located inside the cell, either in the cytoplasm or within specific organelles. Alternatively, some enzymes are extracellular, so they're found outside the cell, and these can be soluble or bound to the cell wall. If we're dealing with plant or animal sources, it's important to identify which organ or tissue produces the enzyme we're interested in. That helps us understand the enzyme's natural context and potential extraction points. But of course, there are other factors we need to consider when choosing an enzyme source. For example, the stage of the organism's life cycle or growth phase when the enzyme production is at its peak. Knowing this helps optimize yield. We also need to think about the equipment and extraction apparatus available. Some enzymes require specific conditions or tools for extraction, which can influence our choices. For large-scale production, the availability of sources becomes critical. We need enough raw material to meet demand. And finally, processing costs are a key consideration, especially if we're planning to scale up for industrial applications. The cost efficiency of the extraction process can make or break the feasibility of producing certain enzymes on a large scale. All these factors together guide us in selecting the most practical and efficient way to obtain the enzymes we need for our purposes. So the question here is, to what level does an enzyme need to be purified? Well, the answer really depends on what you plan to use the enzyme for. The level of purity required can vary quite a bit. For some applications, like in the detergent industry, you don't need a completely pure enzyme. Usually, a partially purified enzyme works just fine because the goal is more about functionality and cost-effectiveness rather than absolute purity. On the other hand, if you're working in medical or pharmaceutical fields, the standards are much higher. Here, you typically require an enzyme that has been purified to homogeneity, meaning it's as pure as possible. This is crucial to ensure safety, efficacy, and to avoid any unwanted side effects. So ultimately, the degree of purification aligns closely with the specific application and its requirements. It's a balancing act, achieving enough purity to meet the purpose without unnecessary steps that could increase cost or time. Now on this slide, we're looking at different ways to separate objects, and there are several key factors to consider. First, you'll notice the numbers listed. These represent different properties like size, density, shape, and even how objects move or settle under certain conditions. For example, objects with different rolling speeds, such as a stone versus a piece of cotton, will behave differently when rolled or moved, which can help us separate them. Similarly, sedimentation, how quickly objects settle in a fluid, depends heavily on their density and size. Then we have sieving, which is a common method for separating particles based on size. Larger particles stay on top, while smaller ones pass through the sieve. These properties, size, density, and shape, are all crucial in choosing the right separation technique. For instance, stones, wood, and cotton differ in all these aspects: size, density, and shape. By understanding these differences, we can select the most effective method to sort or separate these objects. Whether it's by exploiting their shape, size, or how they behave physically, these principles are fundamental to effective separation in many scientific and industrial processes. Let's take a look at the various methods used in enzyme and protein purification, starting with affinity chromatography. This technique relies on specific interactions between the target protein and a ligand attached to the chromatography support, making it highly selective. Next, hydroxyapatite chromatography separates proteins based on their affinity for calcium phosphate, often used for separating nucleic acids and proteins. Moving on to reverse phase chromatography, which separates molecules based on their hydrophobicity, and hydrophobic interaction chromatography, or HIC, that exploits differences in hydrophobic regions of proteins. Salting out is another classic method, where high salt concentrations reduce protein solubility, causing them to precipitate out of solution. Ion exchange chromatography separates proteins according to their charge, while chromatofocusing uses pH gradients to isolate proteins based on their isoelectric points. Disc page, or denaturing polyacrylamide gel electrophoresis, allows us to analyze protein size under denaturing conditions, whereas isoelectric focusing separates proteins based on their charge at specific pH levels. Gel filtration, also known as size exclusion chromatography, sorts proteins by size, and SDS-page provides a way to determine the molecular weight of proteins by denaturing them and running them through a gel. Ultrafiltration is a membrane-based technique used to concentrate or desalt protein solutions. Understanding what we are purifying is also essential. Proteins can be separated from other cellular components like cell debris, small molecules such as amino acids, sugars, nucleotides, lipids, carbohydrates, and nucleic acids. These are all considered macromolecules or small molecules that differ in properties like affinity, polarity, charge, and size. Homogenization is the first step, breaking open cells to release their contents, including organelles and proteins. We often use ammonium sulfate fractionation, a classical method that exploits differences in solubility to selectively precipitate proteins. All these techniques are grounded in the basic principles of enzyme and protein purification, which aim to isolate the target enzyme or protein as pure as possible while maintaining its activity. Each method leverages a different property, be it affinity, polarity, charge, or size, to achieve this goal and to understand the fundamental properties of the biomolecules we're studying. Many biological processes require a purification scheme to reduce the fermentation broth to its pure final product. It's essential because, in complex mixtures, impurities can interfere with the activity and analysis of the enzyme. Studying enzymes in a simple system, meaning only small ions, buffer molecules, cofactors, and the enzyme itself, is really important. It helps us understand their structure, how they work, their mechanisms, how they're regulated, and their specific roles within a larger, more complex system. Additionally, isolating pure enzymes is crucial when we want to use them for specific medical or industrial purposes. Purity ensures that the enzyme performs consistently and effectively, whether it's in a therapeutic setting or in manufacturing. So, in summary, why do we isolate and purify enzymes? To better understand their fundamental properties and to harness their potential in practical applications, ensuring precision and efficiency in both research and industry. Let's talk about extraction, which is a crucial step in our process. Extraction involves retrieving the key data or insights from a larger dataset or source. It's like sifting through a lot of information to find the most valuable pieces that will inform our decisions. The main point here is that effective extraction ensures we focus on what truly matters, saving time and increasing accuracy. Whether we're extracting data from documents, databases, or other sources, the goal is to do it efficiently and accurately. This step connects directly to our overall theme of optimizing workflows and making data-driven decisions. If we miss or mishandle extraction, the rest of our process can be compromised, so it's fundamental to get it right. Remember, the quality of what we extract sets the foundation for everything that follows, so paying close attention here is essential for success. Today, I want to talk about the different extraction processes we can use to obtain desired compounds. First, there are mechanical methods, which involve physical forces. One common technique is using a high-speed homogenizer, where we break down cells or tissues rapidly to release their contents. Another mechanical approach is high-speed ball milling, where grinding media physically disrupts the material, making extraction more efficient. On the other hand, we have non-mechanical methods, like cell lysis, which involve chemical or enzymatic means to break open cells without applying physical force. Cell lysis can be very effective, especially when gentle or specific extraction is needed. Connecting this to our overall theme, choosing the right extraction process depends on the type of material we're working with and the nature of the compounds we're after. Mechanical methods are often faster and suitable for large-scale operations, while non-mechanical ones can be more selective and gentle. Understanding these options allows us to optimize our extraction techniques for better yield and purity. Now, let's talk about high-pressure homogenizers. In this process, cells are subjected to a combination of impact, shear forces, and a significant pressure drop across the valve. The key idea here is that the main disruptive factor is the pressure applied, along with the pressure drop that occurs as the material passes through the valve. This sudden change in pressure creates impact and shear stress, which are proportional to the operating pressure. Basically, the higher the pressure, the greater the stress on the cells, leading to their disruption. This disruption is crucial because it releases the enzymes contained within the cells. So, in essence, high-pressure homogenizers use controlled pressure and shear forces to break open cells efficiently, facilitating enzyme extraction through mechanical means. This method is a vital part of the overall process of enzyme recovery, making it a very effective mechanical extraction technique. Now let's talk about ball mills. When cell suspensions are agitated with small steel or glass beads, usually between 0.2 and 1 millimeters in diameter, they get broken apart through a combination of high liquid shear gradients and collisions with the beads. This process is quite effective for disrupting cells. The rate and how effectively enzymes are released can actually be adjusted by changing a few parameters: the agitation speed, the size of the beads, and even the dimensions of the equipment itself. So, it's a flexible method that can be tailored to specific needs. It's important to note that bead milling can be used on any type of biomass, whether filamentous or unicellular. However, generally speaking, larger cells tend to break more easily than smaller bacteria because of their size and structural differences. This technique falls under mechanical extraction methods, which are quite useful when we're aiming to access intracellular components efficiently. Overall, bead milling is a versatile and scalable option for disrupting a wide range of biomass types in bioprocessing. Let's take a closer look at this slide and explore the key points. First, it's important to understand that the main idea here is about connecting the dots between our strategies and the overall goals of the project. We want to ensure that every step we take aligns with the bigger picture, creating a cohesive pathway forward. Next, I'd emphasize the importance of thorough communication. Making sure everyone is on the same page helps prevent misunderstandings and keeps the team motivated and focused. It's not just about individual tasks, but about how each piece fits into the larger puzzle. Finally, I want to highlight the value of adaptability. As we progress, being flexible and open to adjustments allows us to respond to new challenges or opportunities swiftly. This dynamic approach supports the overall theme of strategic agility, which is crucial for success. By keeping these points in mind, alignment, communication, and adaptability, we're better equipped to achieve our objectives efficiently and effectively. Let's talk about breaking cells using non-mechanical methods. This approach is particularly attractive because it allows us to release enzymes under very gentle conditions. Unlike mechanical methods, these techniques don't subject the enzymes to heat or shear forces, which can sometimes damage them. Plus, many of these methods are quite cost-effective and operate quietly, making them practical for various settings. Some of the available methods include osmotic shock, ultrasonic cell disruption, freezing followed by thawing, cold shock, desiccation, enzymic lysis, and chemical lysis. Each of these has its own advantages and limitations, so choosing the right method depends on your specific circumstances and the type of cells you're working with. While none of these methods are perfect, they can be particularly useful when you need a gentle, inexpensive, and quiet way to lyse cells without compromising enzyme activity. Understanding these options helps us optimize our processes and tailor our approach depending on the situation. All right, now let's talk about osmotic shock. This occurs when there's a sudden change in osmotic pressure that impacts cells. Basically, cells are first exposed to either a high or low salt concentration, which causes water to move in or out of the cells gradually. But the real problem happens when the environment suddenly switches to the opposite condition. For example, if a cell is in a high salt environment and suddenly the salt concentration drops, water rushes into the cell rapidly. Conversely, if a cell is in a low salt environment and then suddenly exposed to high salt, water quickly leaves the cell. This abrupt shift in osmotic pressure causes the cell to swell or shrink quickly, leading to cell lysis or bursting. This process, known as osmotic shock, highlights how sensitive cells are to rapid environmental changes. It emphasizes the importance of maintaining osmotic balance for cell integrity and function, which ties into our broader theme of cell homeostasis. Understanding osmotic shock is crucial in many biological and medical contexts, especially when considering cell preservation or the effects of certain drugs or treatments. Today, I'll explain how ultrasonic treatment affects microbial cells in suspension. When we use inaudible ultrasound, typically above about 18 kHz, it can actually inactivate and disrupt these cells. Now, how does that work? Ultrasonication involves a probe that moves rapidly back and forth in the liquid, creating high-frequency vibrations. These vibrations lead to the formation of tiny bubbles that quickly collapse or burst. When these bubbles collapse, they convert the sonic energy into mechanical energy. Think of shock waves, which can reach pressures of several thousand atmospheres, around 300 MPa. This intense energy creates powerful motions within the cells. If these motions, what we call kinetic energy, are strong enough to overcome the cell wall's strength, they cause the cells to disintegrate. Essentially, ultrasonic cell disruption is a way to break open cells efficiently, which can be useful in various applications like extracting cellular contents or controlling microbial contamination. So, in summary, ultrasonication uses rapid vibrations and bubble collapse to generate shock waves that physically break apart microbial cells, making it a valuable tool in both research and industry. Ultrasonication produces cavitation phenomena when acoustic power inputs are sufficiently high to allow the multiple production of microbubbles at nucleation sites in the fluid. What this means is that as we increase the power, tiny bubbles start to form repeatedly at specific points in the liquid. These bubbles then grow during the rarefying phase of the sound wave. That's the part where the pressure drops, and then they collapse during the compression phase, when the pressure rises again. This collapse is really significant because it creates a violent shock wave that passes through the medium, and that's what makes cavitation so powerful. Essentially, cavitation is the process where gas bubbles nucleate, grow, and then violently collapse due to intense sound waves. This process is fundamental in ultrasonic cell disruption because the shear stress generated by these collapsing bubbles damages cell walls and membranes. So, in the context of our overall theme, cavitation is a key mechanism by which ultrasonication can effectively break cells apart, making it a valuable tool for applications like extraction, sterilization, or even breaking down biological tissues. It's this intense, localized energy release that enables us to achieve such efficient cell disruption. So, freeze-thaw cycles work by a fascinating process. When you freeze the material, ice crystals form inside the cells, which can cause physical damage. Then, upon thawing, these crystals melt and the cells expand, often leading to rupture. This cycle is particularly effective for breaking down algae and soft plant material, making it useful in certain applications. However, one important thing to keep in mind is that this method can be quite time-consuming, since each freeze and thaw cycle takes time to complete. Overall, it's a technique that leverages the physical stresses of ice formation and melting to disrupt cell integrity, fitting into our broader discussion of cell disruption methods. Now, let's talk about desiccation as a method for cell disruption. This technique involves using air drying to damage the cells, and it's typically followed by solvent extraction to isolate the enzymes. It's a straightforward physical method that can be effective in certain contexts. Next, we have enzymatic lysis. This involves adding specific enzymes to break open the cells. While this method is widely used in the laboratory because of its precision, it's less common in industrial applications mainly due to cost considerations. For example, lysozyme, which is derived from hen egg white, is the only enzyme available commercially at scale for this purpose. The main drawback here is that enzymes like lysozyme can be quite expensive, which limits their large-scale use. Moving on to chemical lysis, this approach uses acids, alkalis, surfactants, or solvents to break open cells. The key is that the enzymes or cellular components need to be robust enough to withstand these harsh conditions. Detergents like Triton X100, either alone or combined with chaotropic agents such as guanidine HCl, are particularly effective at releasing membrane-bound enzymes. However, it's important to keep in mind that these chemical agents tend to be costly and can be challenging to remove from the final product, which can complicate downstream processing. Overall, each method has its pros and cons, and selecting the right one depends on balancing efficiency, cost, and the specific requirements of the process. Some types of cells can be caused to lyse simply by exposing them to osmotic shock. This method is actually quite inexpensive, gentle, and convenient for releasing enzymes. However, despite these advantages, it hasn't been widely adopted on a large scale, mainly because it's less efficient compared to other methods. Another approach is autolysis, where certain cells, like yeast, can break down themselves. For example, yeast invertase preparations, which are used in industrial invert sugar production, are produced through this self-destruction process. While autolysis is slower than mechanical methods, it can still be very useful, especially when dealing with large-scale operations. Of course, autolysis does have some potential drawbacks, such as the risk of microbial contamination and the fact that it takes more time. But if speed isn't the primary concern, it can be an effective large-scale strategy. Finally, desiccation, or drying, can also be a very useful technique for preparing enzymes on a large scale, especially when the drying process is carefully controlled. In such cases, slow drying methods are preferred over rapid ones like lyophilization, because they help preserve enzyme activity during the process. All these methods, osmotic shock, autolysis, and desiccation, highlight different ways we can approach enzyme extraction and stabilization, fitting into the broader theme of optimizing enzyme production techniques. Now, when it comes to extracting enzymes, one of the critical steps is solid-liquid separation. This process is essential because it helps us isolate the enzyme from the solid materials after the reaction or extraction process. Basically, after we've done our extraction, we need to separate the liquid containing our enzyme from the solid residues. There are a couple of main techniques we typically use for this separation. The first is centrifugation, which uses rapid spinning to make the solid particles settle at the bottom, leaving the liquid on top. It's very efficient, especially when dealing with fine particles or complex mixtures. The second common method is filtration, where we pass the mixture through a filter medium that traps the solids while allowing the liquid to pass through. Filtration is often simpler and more straightforward, especially for larger particles or when working with less viscous solutions. Both methods are crucial tools in our toolbox, and choosing between them depends on the specific properties of the mixture and the scale of the operation. Overall, solid-liquid separation ensures we can purify and recover the enzyme effectively, which is key to the overall success of enzyme extraction processes. Centrifugation is a powerful technique that separates particles based on their size and density differences. Essentially, when we spin a mixture at high speeds, the denser and larger particles tend to move outward more quickly, settling at the bottom or forming a separate layer. This process leverages the differences between the liquid and solid phases, making it especially useful in various applications like laboratory analyses and industrial processes. By understanding how particles behave under centrifugal force, we can efficiently isolate specific components from complex mixtures. Overall, centrifugation is a fundamental method that highlights how physical properties like size and density can be used to achieve separation effectively. Now let's take a closer look at what happens inside the centrifuge tube before and after centrifugation. On this slide, you can see a sample in the tube initially, where the components are still mixed together. After centrifugation, you notice a clear separation. The pellet, which is the solid material that has gathered at the bottom, and the supernatant, the liquid that remains on top. This separation is crucial because it allows us to isolate specific parts of the sample for further analysis or processing. The formation of the pellet and supernatant is a key concept in centrifugation, as it illustrates how we can effectively separate different components based on their density. Understanding this separation helps us grasp how centrifugation is used in various applications, from clinical labs to research settings. Let's take a look at two common types of centrifuges used in laboratory separations. First, the tubular bowl centrifuge. This type is particularly useful when we need to perform solid-liquid separation, especially in enzymatic isolation processes. It can achieve excellent separation of microbial cells, as well as animal, plant, and most microbial cell debris suspended in a solution. This makes it a versatile tool for various biological separations. Moving on to the disc bowl centrifuge, which is widely used for removing cells and animal debris from samples. Its design allows it to effectively separate these larger particles. Interestingly, it can also partially recover microbial cell debris and protein precipitates, which can be important depending on the downstream application. Both centrifuge types are integral to our overall process because they help us isolate and purify specific components efficiently. The choice between them depends on the nature of the sample and the specific separation goals. Understanding these distinctions allows us to optimize our centrifugation steps and improve the quality of our results. Now, let's look at some specific types of centrifuges used for specialized separation tasks. First, we have the perforate bowl basket centrifuge. This type is particularly useful when separating adsorbents like cellulose and agarose. It handles these materials efficiently by perforating the bowl to allow continuous removal during operation. This makes it ideal for processes where you need to separate or recover these substances effectively. Next, we come to the zonal ultracentrifuge. This instrument is especially important in the vaccine industry because it can easily remove cell debris from viruses, ensuring higher purity of the viral particles. It also has the ability to collect fine protein precipitates, which is crucial in many protein purification protocols. Additionally, the zonal ultracentrifuge has been used experimentally to purify RNA polymerase and very fine debris in enzyme preparations, showcasing its versatility and precision. Overall, these specialized centrifuges expand our capabilities in separating complex mixtures, whether it's removing cell debris from viruses or separating small, delicate particles during purification. They highlight the importance of selecting the right centrifuge type for each specific application to achieve optimal results. Let's talk about filtration. Essentially, filters use a cloth or some kind of porous material, and when you apply pressure, it helps push smaller particles through the filter. This process allows us to separate different elements within a solution based on their size. So, larger particles stay on one side, while the smaller ones pass through. This technique is really important in many laboratory and industrial processes because it helps us clarify solutions or isolate specific components efficiently. Remember, the key idea here is that by using a filter and applying pressure, we can effectively separate particles by size, making filtration a fundamental method in science and manufacturing. Let's talk about batch filtration. This process is usually performed under constant pressure, typically with a pump that moves the broth or liquor through the filter. As the filtration proceeds, a filter cake will begin to build up on the filter surface. This buildup causes resistance to the flow of the broth to increase over time. In industrial settings, the most common equipment used is the filter press. This is essentially a large-scale version of a batch vacuum filter, arranged with plates and frames to hold the filter medium. Batch filtration is quite effective for removing cells from a mixture, but it's important to note that it doesn't work particularly well for removing animal cell debris or plant seed debris. These types of debris tend to be more challenging to filter out using this method. Overall, understanding the mechanics and limitations of batch filtration helps us choose the right process for specific applications in bioprocessing. Now, let's take a look at rotary drum filtration. The solution is vacuumed upward as it crosses a filter septum, which is then removed by a positive displacement pump. This process allows for continuous filtration without interruption. After each rotation, the filter cake is removed, revealing a fresh surface for the next cycle of filtration. This method is particularly efficient for removing substances like mycelia, cells, proteins, and enzymes. However, to enhance filtration efficiency, a filter aid or a pre-coat of the septum might be necessary. Moving on to ultrafiltration, this technique uses a membrane to separate particles based on size. Essentially, it targets particles that are much larger than the solvent molecules. The successful removal typically occurs within the size range of about 10 solvent molecular diameters up to 0.5 micrometers. This makes ultrafiltration particularly useful when you need to isolate or concentrate larger molecules or particles from a solution, fitting into the broader context of our filtration strategies. Let's take a closer look at the techniques, methods, and samples used for cell disruption. Starting with the gentle methods, these are typically used when we want to preserve as much of the cellular components as possible. For example, osmotic shock through cell lysis, enzyme digestion, hand homogenizers, and chemical solubilization or autolysis. These methods are suitable for samples like gram-negative bacteria, animal cells, yeast, and bacteria, depending on the specific technique. Moving on to the mincing approach, this is a bit more manual but effective. It's often used for gram-negative bacteria and animal cells, or yeast and bacteria samples, especially when dealing with tissues like muscle from animals. Then we have the moderate techniques, which include blade homogenizers and grinding with a pestle and mortar. These are commonly used for muscle tissues from animals or plants, as well as bacteria and plant tissues. They provide a good balance between gentle and vigorous methods. Finally, the vigorous methods, such as French press, ultrasonication, bead mills, and Manton-Gallon homogenizers, are used when we need to break open tougher samples like bacteria, yeast, isolated plant cells, or microbial suspensions. These methods are powerful and ensure thorough disruption when gentler techniques might not suffice. Overall, understanding the right technique, method, and sample combination is essential for efficient cell disruption, which is a fundamental step in many biotechnological applications. Let's take a closer look at some of the practical considerations when working with enzymes. First, heat is a major factor. All mechanical methods generate heat because they require a significant input of energy, and this heat can affect enzyme stability. That's why cooling is so essential during processing. Additionally, the presence of substrates, substrate analogs, or polyols can help stabilize the enzyme, making sure it remains active and effective. Moving on to shear forces, these are necessary to disrupt cells, but they can also cause damage to the enzymes themselves. The risk of damage increases if heavy metal ions or air interfaces are involved, so controlling shear is important. Now, regarding proteases, breaking open cells releases enzymes that could degrade your target enzymes, leading to activity loss. To minimize this, increasing processing speed and cooling helps. Also, adding excess substrates like inexpensive proteins or specific inhibitors can protect the enzymes during extraction. pH is another critical factor. Using buffered solutions helps maintain an optimal pH environment, and again, substrates or polyols can contribute to enzyme stabilization in these conditions. Chemical interactions also matter. Some enzymes are sensitive to detergents or solvents, which may cause conformational changes. Plant-derived polyphenolics are potent enzyme inhibitors, but this challenge can be addressed by using adsorbents like polyvinylpyrrolidone or reducing agents, such as ascorbic acid, to inhibit polyphenol activity. Oxidation is another concern, and incorporating reducing agents like ascorbic acid, mercaptothenol, or dithiophthalate can help protect enzymes from oxidative damage. Foaming poses a unique challenge, as the gas-liquid interfaces in foam can disrupt enzyme structure. Managing foam formation is thus important during processing. Finally, heavy metal toxicity is something to watch out for. Heavy metal ions like iron, copper, or nickel can leach into your samples from equipment. Using chelating agents such as EDTA can effectively bind these metals and protect your enzymes from irreversible inactivation. All these factors highlight how careful control of the processing environment is crucial to maintaining enzyme activity and ensuring successful outcomes. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. Now, let's talk about why it's so important to purify industrial enzymes. Essentially, purification helps us remove any foreign substances that might inhibit the enzyme's action or interfere with the overall process. Think of it like cleaning a lens to get a clearer picture. When enzymes are purified, their activity increases, which directly improves the efficiency of the process we're working on. It's also crucial to understand that the level of purity required really depends on the specific application. For instance, pharmaceuticals demand very high purity, while other industrial uses might be more flexible. The methods we choose for purification and separation are generally based on properties like solubility, size, polarity, and how strongly the enzyme binds to other molecules. When selecting a purification method, we need to consider several factors, including the scale of production, the timeline, and the specific properties of the enzymes involved. All these elements influence which separation technique will be most effective and practical for our needs. So, in essence, purification isn't just about cleaning up the enzyme. It's about tailoring the process to fit the application and ensuring we get the best possible outcome. The principle behind this type of separation is that enzyme solubility can change dramatically when factors like pH, ionic strength, or dielectric constant are altered. This change often leads to the formation of precipitates. Precipitation, in essence, is a process where adding an ionic solution to an ionic fermentation broth causes insoluble particles to form. For example, most proteins become less soluble at high salt concentrations, a phenomenon known as salting out. It's important to note that the salt concentration at which a protein precipitates varies from one protein to another, which allows us to use salting out as a way to selectively fractionate proteins. We can also induce precipitation by adding water-miscible organic solvents such as ethanol or acetone. These solvents change the dielectric constant of the solution, which can cause the target enzyme to precipitate out. Additionally, neutral water-soluble polymers can be used for this purpose instead of organic solvents. However, we must be cautious because there's a risk of losing enzyme activity during the precipitation process. Plus, we need to consider how to further separate the enzyme from the added salt or polymer afterward. Overall, solubility-based separation is a powerful technique, but it requires careful optimization to balance enzyme recovery and activity retention. Let's take a look at how temperature and pH variations can influence protein and enzyme solubility. Generally, most proteins and enzymes become more soluble as the temperature increases. However, it's important to be cautious because too high a temperature can lead to denaturation or loss of the product. Now, regarding pH adjustments, by changing the pH, we can alter an enzyme's polarity. When we reach a pH where the enzyme has a zero net charge, meaning its polarity is at its lowest, its solubility in water drops significantly. This is a useful trick for precipitating enzymes selectively. Moving on to precipitation using organic solvents. When we add an organic solvent to an aqueous fermentation broth, the dielectric constant of the solution decreases. This change causes the solubility of proteins and enzymes to drop, leading them to precipitate out. This method is quite popular industrially because it's cost-effective and straightforward to implement. Finally, let's talk about using metal ions for precipitation. Certain metal salts, such as magnesium chloride, can form insoluble complexes with proteins and enzymes, effectively precipitating them out of solution. But it's crucial to remove nucleic acids beforehand, since they can interfere with this process and reduce separation clarity. Manganese salts, for instance, can be used to selectively precipitate nucleic acids, helping us clean up the mixture before removing the proteins or enzymes we're interested in. You can also precipitate the enzyme using coagulation or flocculation techniques. Now, coagulation involves particles directly adhering to each other, which helps in aggregating impurities or enzymes. Flocculation, on the other hand, uses an agent that acts as a bridge linking particles together into larger flocks. This process makes it easier to separate or recover the enzyme from the mixture. Both methods are useful tools in enzyme recovery, and choosing the right one depends on the specific characteristics of your process and the nature of the particles involved. These techniques are important because they enhance the efficiency of enzyme precipitation, ultimately improving the overall process. Since enzymes are relatively large molecules, separation based on size or mass is an effective way to purify them, especially those with high molecular weight. One common method we use is dialysis, which involves semi-permeable membranes that allow small molecules like salts, organic compounds, and peptides to pass through, effectively removing them from the enzyme solution. Filtration methods are also very useful for further purification. Techniques such as ultrafiltration, cross-flow filtration, and tangential flow filtration are designed to separate proteins based on their size. Similarly, size exclusion chromatography, also known as gel filtration, separates enzymes by their size as they pass through a porous matrix. It's really important to know the size of your enzyme when choosing these methods. Selecting the appropriate pore size for filters or dialysis membranes ensures you effectively separate the enzyme from smaller contaminants without losing the enzyme itself. Overall, these size or mass-based methods are powerful tools in enzyme purification because they exploit the size differences, helping us obtain highly purified enzymes for research or industrial applications. Let's look at this example of SEC, or Securities and Exchange Commission, to better understand how it functions. The SEC plays a crucial role in overseeing securities markets, ensuring transparency and protecting investors. Here, we see how it enforces regulations and maintains fair trading practices. This example highlights the importance of regulatory agencies in maintaining market integrity, which connects directly to our overall theme of financial oversight and investor confidence. It's a practical illustration of the principles we've discussed about regulation and compliance in financial markets. Enzymes can be separated based on their polarity, which essentially relates to their net charge, charge density, and hydrophobic interactions. These properties influence how enzymes interact with different separation mediums. One common method is ion exchange chromatography, where we use a column filled with beads that have either negative or positive functional groups. By passing a mixture through this column, enzymes with opposite charges are attracted and can be separated effectively. For example, cationic enzymes are separated on anionic columns, while anionic enzymes are captured on cationic columns. Another technique is electrophoresis, which uses an electrical field to move ions through a solid or semi-solid matrix. This method separates components based on their charge density, with more highly charged molecules migrating faster. The most widely used form of electrophoresis is SDS-phage, or sodium dodecyl sulfate polyacrylamide gel electrophoresis. It's a powerful way to analyze enzyme purity and size. Then we have hydrophobic interaction chromatography, or HIC, which relies on differences in hydrophobicity. Enzymes are adsorbed onto matrices like octyl or phenylcephalose, which have hydrophobic groups. By gradually decreasing ionic strength or increasing nonpolar solvents, we can elute the enzymes, usually obtaining fractions with high purity. This technique is similar in principle to high-performance liquid chromatography, or HPLC, and is useful for separating enzymes based on their polarity and hydrophobicity. Overall, these methods, ion exchange, electrophoresis, and hydrophobic interaction chromatography, highlight the importance of polarity and charge in enzyme separation, helping us isolate specific enzymes for further study or application. This technique takes advantage of the high affinity that many enzymes have for specific chemical groups. It's a clever way to isolate proteins based on their unique binding properties. The most common method used here is affinity chromatography, which leverages these specific interactions. Essentially, proteins are separated according to their binding specificity to a ligand attached to the chromatography matrix. Proteins that do not bind to the ligand will simply wash out of the column, leaving behind those that have a strong affinity. This method is highly selective and efficient, making it a powerful tool in protein purification workflows. It really highlights how understanding enzyme specificity can be practically applied to isolate target proteins effectively, tying into the broader theme of targeted purification techniques. Aqueous two-phase systems, or ATPS, are fascinating liquid-liquid phase separation systems that are increasingly used for enzyme purification. These systems consist of two immiscible aqueous phases, which are created by mixing two water-soluble polymers at specific ratios and pH levels. Common polymers used include polyethylene glycol, or PEG, and dextrin. The core idea here is that enzymes can be selectively partitioned into one of these two phases based on their unique physicochemical properties, such as size, charge, and hydrophobicity. What's really interesting is that we can influence this partitioning behavior by adjusting the composition of the ATPS, things like polymer concentration, pH, salt levels, and temperature. This flexibility allows us to optimize the purification process, making it more efficient and selective. Overall, ATPS provides a gentle, scalable, and environmentally friendly way to purify enzymes, aligning well with the broader theme of improving bioprocessing techniques. Now, let's talk about how we can purify enzymes using aqueous two-phase systems, or ATPS. The process begins by mixing your enzyme-containing sample with the ATPS. This mixture allows the enzyme to partition into one of the two phases based on its properties. Once the enzyme has separated, we can use centrifugation or other separation methods to isolate the phase that contains the enzyme. From there, further purification steps like chromatography or dialysis can be employed to achieve the desired purity. One of the biggest advantages of using ATPS is that it's a gentle method. It doesn't denature or damage the enzyme, so the activity and stability are preserved. Plus, it's scalable, making it suitable for large-scale purification processes. And the best part is that it works for a wide variety of enzymes with different characteristics. Overall, ATPS offers a versatile, efficient, and enzyme-friendly approach, which ties nicely into our broader theme of optimizing enzyme purification techniques. Now, let's talk about enzyme purification using chromatography techniques. Chromatography is a powerful method that allows us to separate and isolate specific enzymes from complex mixtures. The key idea here is that different molecules have unique properties, such as charge, size, or affinity for certain materials, which we can exploit to achieve separation. In practice, we pass our mixture through a chromatography column containing a specialized medium. Depending on the type of chromatography, whether it's ion exchange, gel filtration, or affinity chromatography, different enzymes will interact with the medium in distinct ways. This interaction is what enables us to separate our target enzyme from others. The most important concept to grasp is that chromatography provides both high specificity and efficiency, making it ideal for obtaining pure enzymes needed for research or industrial applications. And as we've seen throughout this presentation, purifying enzymes is a critical step in understanding their functions and harnessing their potential. Ultimately, chromatography is a cornerstone technique that ties into our broader theme of enzyme analysis and application, helping us unlock the full potential of these biological catalysts. Let's take a moment to really emphasize the key points here. First, understanding these concepts thoroughly is crucial because they form the foundation of what we've been discussing throughout the presentation. It's not just about memorizing facts, but about grasping how these ideas connect and influence the bigger picture. I want to highlight that the most important concepts we've covered are interconnected, and recognizing those links helps us see the full scope of the topic. Remember, this slide ties back to our overall theme of understanding the core principles that drive success in this field. So, keep these connections in mind as we move forward, and think about how applying these ideas can make a real difference in practical scenarios. Now, let's move on to enzyme purification using the filtration technique. This method is quite straightforward, but very effective in separating enzymes from other components in a mixture. The basic idea is to use a filter that allows smaller molecules, like water and salts, to pass through while retaining larger molecules such as enzymes. This way, we can concentrate and partially purify the enzyme. It's particularly useful when dealing with large volumes or crude extracts. The key points to remember are that filtration is a gentle process, which helps preserve enzyme activity, and that selecting the right pore size is crucial for effective separation. Overall, filtration is a fundamental step in enzyme purification because it's simple, cost-effective, and preserves enzyme functionality, integral to our broader goal of obtaining pure enzymes for various applications. Let's walk through an example of how to purify DNA polymerase, which is a crucial step in ensuring enzyme activity and purity for our experiments. First, we'll start with the source material containing the enzyme, usually from bacterial cells or other organisms that produce it naturally. The goal here is to isolate the DNA polymerase from all the other cellular components. We typically begin with cell lysis to break open the cells and release the proteins. After that, we use a series of purification steps, such as chromatography techniques. These might include ion exchange, affinity, or gel filtration chromatography. Each step helps to separate the DNA polymerase based on its specific properties, like charge or size. During purification, it's important to monitor the activity of the enzyme and check for contamination. We often do this with assay tests that measure the enzyme's ability to synthesize DNA. Also, maintaining the right conditions, like pH, temperature, and salt concentration, is vital to preserve the enzyme's activity throughout the process. Finally, after multiple purification steps, we end up with highly purified DNA polymerase, ready for use in PCR or other molecular biology applications. The key takeaway here is that purification isn't just about removing impurities. It's about optimizing the enzyme's activity and stability, which ultimately impacts the success of your experiments. This process exemplifies how careful technique and understanding of protein properties are essential in molecular biology workflows. Terima kasih banyak atas perhatian dan waktu yang telah kalian berikan selama presentasi ini. Saya sangat menghargai kesempatan untuk berbagi informasi dan wawasan dengan kalian semua hari ini. Jangan ragu untuk mengunjungi website kami di www.upam.edu. May untuk mendapatkan lebih banyak sumber daya dan informasi terkait topik yang telah kita bahas. Semoga apa yang telah disampaikan hari ini dapat memberikan manfaat dan inspirasi bagi kalian semua. Sekali lagi, terima kasih atas kehadiran dan partisipasi kalian. Sampai jumpa dan semoga hari kalian menyenangkan.